All right, well, I think most everyone is here. I think a few people are still coming in. That's okay, but we'll go ahead and get started. Um, well, welcome to the last event of Research and Internship Day. This is the keynote speech, and we're so excited to have Dr. Bottazzi here with us today. Thank you everyone for attending all of the events throughout the day, and I hope you guys learned something today from Dr. Bottazzi's research. So I'll go ahead and hand it over to Annie now.
Thank you. Yes, thank you so much, everyone, for making it. I know we had a bit of a technical issue, of course, but thank you for working with us and for being patient. So now I'm going to introduce Dr. Bottazzi. We have Dr. Maria Bottazzi for our keynote speech today at Research and Internship Day. She is a professor of pediatrics and the Associate Dean of the National School of Tropical Medicine at Baylor College of Medicine, as well as an editor-in-chief for Springer's Tropical Medicine Reports. Dr. Bottazzi is also the co-director of Texas Children's Hospital Center for Vaccine Development, where she helps to develop vaccines for neglected tropical diseases and other emerging and infectious diseases. She is also a distinguished professor at Baylor. She is an internationally recognized vaccinologist with more than 2 decades of experience advancing product development partnerships. So please join me all in welcoming Dr. Maria Bottazzi.
Thank you. Thank you so much. It's so nice to see. Unfortunately, my screen can only take a few faces from the 109 that I see that it joined this presentation. So I'm going to try to maybe give you around 20, 30 minutes, maybe 35 minutes of my keynote, but then I'd like to really make it a little bit more interactive, so open to any questions that you may have. Of course, initially, I have to say that I'm very happy and very thankful that you guys invited me. And maybe remind everybody to put yourselves on mute. Sometimes it's hard to remember that. Thanking also Dr. Riz, because she always invites me to very interesting events that you students organize. So I'm very happy to be with you. Let me share my screen, which I think it's this one. Hold on. Sometimes the actual presentation doesn't show up. Hold on. Make sure the presentation is on. There you go. Now. Okay. Now you can see it, right? Full screen? Yep. Okay. Sounds good. I'll minimize your faces so that I can pay attention to my screen. So again, first of all, as a professor at Baylor, including not only Baylor College of Medicine and Baylor University, I think I'm very excited to talk to you about why, I should be the one that I have a bias in telling you why is it important to do research. And certainly nowadays, even more important to not only do the research, but really learn the meaning of what research really means and why is it so important for us to use that as a tool to be innovative and certainly, identify solutions to many of the problems that we have in the world. So I'm going to tell you a little bit about of course, who we are and the type of research that we do. But of course, I'm going to be focusing very much on COVID-19 vaccines, as an example of what, is going on around the world right now and why we think that, of course, they're going to be a very essential and very important tool to try to, go back to some level of normality. But at the same time, why is it important that we continue investing into doing research, especially coronaviruses, so that we can prevent having another situation like we've been seeing for the last 10 months. So I always like to start like very high level, right? Like just looking at what's going on around the world. And to be honest, in the area of global health, it's almost like playing a whack-a-mole game, right? Because, certainly it's very hard for any one group or any one institution or any one even sometimes agency or even country to be able to address all the problems that we have around the world. But, albeit, it's probably a hard task. What is even harder is that sometimes we put a lot of attention maybe in, how to understand and prevent, these diseases that we call communicable diseases, that are, of course, diseases that are infectious. And of course, we like the diseases that we know have a potential to have vaccines, prevent such diseases, or, even have other tools to prevent these diseases. Now, there's been a lot of resurgence and certainly a lot of attention amongst the diseases that are infectious, but that also have vectors that can either be reservoirs or that can be intermediaries and that can transmit these diseases. And one of these also types of infections are these so-called emerging infectious diseases. They call them emerging because they usually live already in another species, and then they kind of like jump over and they emerge as a human disease. But this also reminds you that when we talk about communicable diseases or any kind of zoonotic and vector-borne diseases, these diseases are generally what we call One Health. So we, of course, like to put attention to the humans, but we also have to remember that a lot of these diseases also afflict animals and sometimes even plants, and sometimes even not only vertebrate animals, but also invertebrates. And then you have the other side of the picture, which are the non-communicable diseases, like diabetes, obesity, or high blood pressure, or any of these other chronic diseases. And more we're now seeing this link that most likely there are factors, of course, that influence humans to have all sorts of these chronic non-communicable diseases. But we now see that when you also have infectious diseases, they either can exacerbate and make worse the non-communicable diseases, or that because you have non-communicable chronic diseases, you actually can be more prone to getting an infectious disease. So this is why it's hard to say, certainly I have an expertise primarily in infectious diseases, but when we work in this space, we usually end up having to also be quite well immersed into folding everything and looking at things that are more global. Now, how has the world attempted to tackle this notion of global health issues? And in fact, it really, even though it probably, of course, started way before the year 2000, but something that really was transformational was what happened at the beginning of this century. And it was when, through the United Nations, there was the creation of the so-called Millennium Development Goals, which I have to say that amongst the many initiatives that you arguably can see coming out of governments or agencies or even multinational or global agencies, the MDGs are probably one of the most discussed, one of the most followed, and one of the most aspirations that everybody signed up to try to contribute, meaning that it actually was a very good thing for the world because the countries all pledged to try to abide by what this list of goals would be, with very, very, very specific targets, with, of course, a very specific timeline. And even though arguably you can never really reach them 100%, I mean, there were many, many things that happened during this initial 2000 to 2015. So in 15 years, we saw some enormous gains and learnings just by following these MDGs. And as you can see, back in the early, initial of the century, we were really focusing on 8 main MDGs, and of course, they're very broad, but if you look into each one of them, you can really like hone in and, and go into like very, sub aims. And when 2015 occurred, and of course you can go into the United Nations and look at all the reports and the gains and learnings. There was an aspiration that we wanted to continue such goals. And what actually happened was that they got converted into the global goals, also called the Sustainable Development Goals. Again, another 15-year time period. We went from 8 to 17, and they were a little bit more inclusive. And as you can see, for example, we also include a little bit more overarching goals, which are related to climate and related to, of course, conflict and the oceans. And it still retained a few of the original, but they were made, broader. So this is very important because all of us, or any of us who really are interested in working and of course doing research in the space of health, and certainly in the space of global health, I can assure you that we are tackling either a little piece or a bunch of these global goals. And this is also a really good tool, because when you are trying to create the proposals for you to get funding, or when you're trying to advocate for a solution, if you always refer that is going to make a real impact by addressing one of these goals, generally people tend to pay a little bit more attention. So everything that you do, most likely in your daily life, hopefully, and especially as you train and study, and eventually if you want to go to med school, if you want to do go into a research profession, it's keep an eye on what's happening with these global goals. So I'm going to give you, for example, what happened during this. If we're now 20 years in this century, what happened during this initial 20 years, because there's some key things that also have really provided a lot of opportunities and really provided means for ideally creating what we call an infrastructure, but also a business model of how we then can adapt the goals, but then implement and then operationalize them. And so one of the things that, which of course we were very much involved, was if you actually look back and you look at this Millennium Development Goal number 6, which is called combating HIV/AIDS, malaria, and other diseases, that the term other diseases for us was a little bit of a disappointment, because if you were to ask me to define what an other disease mean, it wasn't really very attractive. I mean, it's not really attractive, as a brand. It's not really attractive to funders. It didn't really have anything that would really clarify what do you really mean by other diseases. And it was very difficult to compete by explaining to people, or compete by convincing funders to give you money, because everything got really very skewed to what we call the big 3 diseases, right? In this case, HIV, AIDS, malaria, and probably also tuberculosis. But the other diseases were kind of like very obscure, and it was very hard to try to gain interest and funding for. So with Dr. Hotes, who of course works with me for the last 20 years, and together with a couple of his colleagues from London, one from the London School of Tropical Medicine and one from Liverpool, they really started doing practically a branding exercise, right? So if any of you are interested in business, or interested in certainly marketing, it was really a marketing exercise. So between 2000 and maybe 2009, 2010, they really moved and shaped the whole scientific community, to the point where now, in fact, other diseases is now co-branded, and that's where that term, of neglected tropical diseases research, where people now call them diseases of poverty, or these neglected infections. So clearly they at least gave a branding to these so-called other diseases, which are, of course, very highly prevalent, very, potentially emerging, re-emerging. And now, in fact, probably nobody even, recalls that, even World Health Organization and big agencies such as the Bill and Melinda Gates Foundation, now, it's a term that everybody already kind of recognizes, even as in the public domain. At the same time that this was occurring, another interesting fact is that exactly in the year 1999-2000, it's when the Bill and Melinda Gates Foundation was created and it was launched. And as part of their business model and as part of their mission, they decided that in order for you to be able to develop any global health technology that would indeed address these big three, right, the HIV, TB, and malaria, plus all these other diseases, it would be very important and key to do it in a way where partnerships had to be the driver, meaning saving resources by working together in collaborations, saving resources by bringing different capabilities. And therefore, these so-called product development partnerships were created. So for example, which now we call ourselves the Texas Children's Center for Vaccine Development, we actually are categorized as a PDP. And we were created with seed funds from the Bill and Melinda Gates Foundation. And now we've been in operation for 20 years. And for these last 10 years, we actually moved from, we used to be in the Washington, DC area. And in 2011, we moved down to Texas. Also allowed us that for research, we brand ourselves as the Texas Children's Center for Vaccine Development. But at the same time, we created now the so-called National School of Tropical Medicine to expand our remit beyond research, but also, of course, doing education and training. And we also do some clinical initiatives, and we do policy, and we do other things. At the same time that this was happening, if not sure if you may recall, but unfortunately there were, the starting of really this resurgence of what we call the bioterrorism, biodefense. There was the time, unfortunately, in fact, right in 2011, when of course we had 9/11, and then we also had the scare of the anthrax scare. Through the National Institutes of Health, also these centers of excellence started to be created in this area of biodefense research. And so the combination of global health, infectious diseases, diseases of pandemic importance, diseases that potentially also could be of biodefense importance for security, that's what led to certainly strengthen the need for doing R&D in this so-called emerging and neglected infections. In addition to that, again, so you have now an ecosystem that was being created, right? So you have the global goals, you have the importance of these diseases, and the recognition that they could be of even security, for countries, importance. We also started seeing how dissemination of information was taking a different trend, right? More transparency, more open access, more ability of sharing the scientific findings. And that's as an example, at the same time, around in the year 2000s, we also spearheaded the Public Library of Science model by opening a journal also in the neglected tropical disease area, where Peter was the founding editor-in-chief. And for example, journals like, Anne introduced me with, which is this current tropical medicine report. So as you can see, a lot of, things were happening during this beginning of the century. So when we then came down to Houston and created this National School of Tropical Medicine, our belief is that, our work is to be fully blown with a mission to try to provide value to lives that, which should be equitable, right? That we have to address this urgent need of these diseases that disproportionately, of course, afflict, the poorest people, because as you know, most of the times access to healthcare, infectious diseases tend to be linked with poverty and other, social determinants. And therefore, we came down here with the goal to try to solve problems using, of course, the scientific horsepower of not only the medical center in Houston, but certainly what we have in Texas. We, of course, do fundamental research, translational research, clinical research, but at the same time, we do a lot of advocacy. And this is why it's important for you all to see why, therefore, we aligned immediately with Baylor University, right? So if you even look at, the mission of your university and the mission of our school, it was immediate. So I think that the moment we arrived, we immediately got ourselves in love with, all of you as students, the Baylor University mission, because we share this, willingness to serve. We, of course, are also very interested in the interdisciplinarity. And even though I'm primarily working very closely with, of course, the College of Arts and Sciences, with Baylor, the pre-health programs, with Dr. Klausmeyer, with, the opportunities that we've set up. We also collaborate with, other schools and departments. And so I'm going to tell you very briefly what we're doing with, two programs, which I think, are appropriate for you all. And of course, then you can talk to Riz about, more in detail. And then I'll go into a little bit of, example of the type of research that we do. So the first one is, again, to try to already give you like a tropical medicine 101 introduction. We now, in fact, we're planning not only to do this during just the summer, because now that we can do some things relatively hybrid or virtual, we expect that we potentially could do this not only during the summer months, but right now we have a so-called Summer Institute, which is an introduction to tropical medicine, which is primarily to familiarize you with, what is global health, what is, what are the infectious diseases. We have a hands-on laboratory, which I hope we can retake next year. This year we had to not do it hands-on, but we had to do it virtually. We also do career counseling, we take field trips, and we usually do this like as an intensive course. We also now extended this where there's an opportunity that, after you take this course, or even in parallel, of taking this course, we offer the opportunity of you coming to our labs and also spending 9 weeks as an intern in our labs. We usually can take not very many of you. The institute is a little bit larger, but the labs, we have to, of course, pair you with the appropriate investigator, and of course have a mini project. And we go more in depth of what it means to do some of the research that we do in our labs, especially, of course, in the area of neglected infectious diseases, vaccines, some diagnostics, and some, of course, even molecular epidemiology. And it's actually very comprehensive, and it really just also gives you a very good introduction of what we mean by, laboratory bench research, and translating that, and even using and the learnings that you already have from your classes, how you really can operationalize it in real life. So again, when we also talk about global health technologies and solutions, of course, we're biased with a very few, but most of the learnings also can be expanded into other type of technologies, right? So, making a vaccine or a drug or a diagnostic, is one aspect, but you also have many other technologies, from even just, software, apps. Nowadays, everything has a lot of artificial intelligence involved. But for that one, remember, is not just understanding the biology or just understanding the scientific underpinnings of the problem or a technology. Is one thing that I really suggest a lot is when especially you all are studying and when you're doing you have a major and then you're also doing a minor in something else, or sometimes you even do double majors, or and Baylor universities has some wonderful opportunities of interdisciplinary. I really like when you all have opportunities to study, for example, biology together with business, biology together with law, linking with engineering, linking with informatics, because at the end of the day, you need this to really fill this ecosystem of doing the research and development, creating the values for your discoveries, and eventually, of course, providing an actual tool that can be used in the community. Okay, so let's now go into vaccines, and let me spend the rest of the talk on what are we doing. So I think everybody, I hope, really shares my belief that vaccines are probably one of the best interventions that in public health, because they can prevent some very serious diseases. Certainly in the pediatric population, but now, of course, we see more and more that vaccines can also prevent diseases in adults and even in the elderly. And in fact, if we look amongst that first 15-year period or so, there's studies that the global burden of disease does to see the difference of how much we have really reduced the burden of these diseases in our children from even just 17 years, right? From the year 2000 to the year 2017, we're really seeing, enormous gaining just by the fact that, for example, the Global Alliance for Vaccines, which is called Gavi, which was created, in fact, it was amongst other things that the foundation, Gates Foundation, created. In addition to the PDPs, they also gave $750 million to create this alliance, because this alliance allowed to bring these vaccines to the developing world, right? And just by being able to expand the use and the distribution, the access, the cost structure, it went to enormous gainings. Now, unfortunately, in the recent in fact, even pre-pandemic, we are now seeing a huge vaccine risk index. And this is one of papers that Peter collaborates with to really identify that there's a lot of factors right now that we're seeing around the world. And in fact, it's been exacerbated now by COVID, but it's been really, it really actually started pre-COVID with, issues about climate change, issues about, poverty, urbanization, deforestation, the fact that we have, of course, political instability and even vaccine hesitancy and even anti-science, that a lot of the gainings that we had achieved because we were really reaching the populations and distributing these vaccines, we are now seeing a lot of loss in those gainings. Examples like, when we have measles outbreaks in the US, I mean, it's unheard of. Why do you think we should have measles outbreaks in the US when we have, readily available vaccines? And again, it's all because of this, probably vaccine anti-science and anti-hesitancy or hesitancy for vaccines. In the context of COVID, this, of course, now has been even exacerbated. And if you want a really interesting read, of course, a plug for Peter, he, of course, published a book called The Blue Marble Health, which now he actually has, and it's open, it's free for you to download. It's an electronic little book, in fact, where he now put in context why COVID-19 has been so terrible, especially in underserved populations, certainly poorest populations around the world, and where, in fact, when we say poor, it's not necessarily poor in countries that are what we consider the low middle income countries, or even just the low income countries. It's in fact that most of the cases and most of the severity, and unfortunately I have to say it's a little bit of shameful that, you know, the United States is the top on the list. And as you can see, you know, these are large economies, and these are in fact the economies that now we have seen even a worse impact of the COVID-19. And again, and it's a combination of many, many, many social determinants, and certainly potentially even political factors. But you know, it's really a tragedy. So I want to shift over and then tell you a little bit of what is then going on with the vaccine development space. And I'm sure you have heard this in the news repeatedly, but I just want to walk you through as simplistic as possibly I can. And that's why I present to you my very simple puzzle, where, as you can see, for you to develop a vaccine, you have to have at least 3 pillars that, you know, then they are glued together by hopefully the fact that we want vaccines that would be affordable, equitable, of course, global. But for you to have affordable, equitable, and global access to vaccines, you clearly have to have production, or vaccines to actually being produced, and even prototypes of the design of the different vaccines. Then you have to take those products and you have to test them and evaluate them and establish their safety and establish their efficacy, right? And then, you know, even before you can certainly distribute them, the whole ecosystem of how you're going to plan their delivery, and their delivery meaning even from, you know, do we have the right freezers or refrigerators? Are we going to have the right people trained to deliver them? How are you going to export them? How are you going to vial them? Are you going to, you know, because by producing vaccines, you're not producing the actual material that goes inside the bottle, but you also need to make sure we have enough bottles, enough syringes, enough caps, right? So this is my triangle of, you know, the simple 3 stages of vaccine development, right? You produce, you test, and then you have to deliver. And the hope is that then, therefore, those that we are able to deliver are delivered in a manner that everybody can afford them, and they can be equitably delivered, and therefore we can achieve global distribution. So I'm going to tell you a little bit about what's going on with the production. What are the platforms to produce? What are the targets that we use? Who are the producers? Who's testing and how they're tested? What are these delivery considerations? And as you know, we have a horse in the game, because we actually do have a vaccine that we're developing, and I'll tell you what we are doing to achieve ultimately the affordability, equitable, and certainly safety and efficacy. So there's many trackers that you can look at. You can look at the New York Times, you can look at WHO, but this one is the Milken Institute tracker. And as of this morning, we have more than 200 vaccines that are in development. Just that number for a single disease, it's unprecedented. It's very rare that you have 200 different groups or 200 different vaccines being developed for a single organism or disease. We now have 35 that are in clinical testing. You would say, you know, why do you need so many? Because honestly, only around maybe 1 to maybe 5% of all of these will actually make it to the end. So more you have at the beginning, more chance that we're going to get at least, hopefully, maybe 2 or 3, maybe half a dozen if we're lucky. Ideally, of course, you want at least 1, but I think we're going to have more than 1. We're going to have a little toolbox of potentially different vaccines that are going to serve probably different purposes. They're going to be rolled out in different stages. So some of them may be first generation and may not be the pitch perfect ones, but then maybe we can have better ones or improved ones or complementary ones as time goes by. These are the categories of how you make them, right? So you can make vaccines in multiple different ways. You can make them by taking the actual pathogen, like the COVID-19 virus, and you can inactivate it, or you can attenuate it, and you can then inject it back into a person. And of course, it's non-pathogenic. You can take a piece of that virus and in the lab genetically engineer a sequence that then you can artificially make a protein, and that's called a subunit. These are the only 3 types of vaccine categories that we know we have been able to make before that have been already licensed as a vaccine that we already use. None of the other technologies, for example, DNA-based vaccines, RNA-based vaccines, or vaccines that are produced by inserting genes into other viruses, in this case, they're called viral vectors, they're all experimental, but we've never had a vaccine license before that used these other categories. So we, for example, decided that we were not going to risk playing with a technology that we still didn't know how suitable or safe or effective it would be as a producer of our vaccine. We opted for using the protein subunit category. But many of the vaccines that you hear, in fact, probably most of the vaccines that you're hearing are using experimental technologies. That doesn't mean that they may not work. It's just that you're adding another level of area of unknown and another level of risk, because at least with these categories, we've seen precedent for them. Of course, not for COVID, but we've seen precedent, for example, the hepatitis B vaccine or any of the measles vaccines or the flu vaccines. They all already use these categories. Now, what do you target for the vaccine? So as you know, a vaccine pretends to already prepare your body to make you practice as if you were to see a pathogen, right? So I immunize you with a piece of a pathogen, in this case, for example, a protein, and I ask your body to make a response against that protein. But that protein doesn't cause any disease, right? But this protein is something that resembles a piece of something that a real pathogen has, such that when you then get infected, you already say, oh, I've seen this before, and I know exactly what I need to do. I'm going to make antibodies, I'm going to make cytokines, I'm going to make T cells, and I already can attack it very rapidly. So the strategy for the coronavirus is that we know that the coronavirus, through these spike proteins, which in fact is actually a key. So the spike is a key, is the piece of the virus that actually gets itself inserted into a receptor, which is the ACE2 receptor on our cells. So if we block the ability of the spike to enter the keyhole, we basically, in theory, should prevent the virus to enter the cell, and therefore the virus cannot replicate, and therefore you don't have disease. So the key for that is that when you mount an immune response, is that you have these so-called antibodies, which are also considered neutralizing, because it neutralizes a function, it blocks a function. And more of these antibodies you have, more specific they are, ideally more you're going to block the virus to infect. And so all of us are trying to, at some level, look at how we're going to make these high levels of neutralizing antibodies, paired it with, of course, a milieu of other things, which are cytokines and, of course, T cells. So knowing that premise, knowing that we have a category of production, and we know that we want to target that either full spike or a piece of the spike, who produces vaccines? And so I think most of you hear that there's very large multinationals, right? Pharmaceutical companies, vaccine companies, Merck and GSK and Pfizer and Johnson & Johnson. But there's also a category that are the network of developing country vaccine manufacturers that maybe you don't recognize their names, but believe it or not, many of the vaccines that are made, especially for the globe, are made by these DCVMNs. They're mostly located in India, mostly located in Asia. There's a few here in Latin America, unfortunately not very many, as you can see from the graph. However, as you can see, still Most of them are in either North America, Europe, and Asia and Australia. So one of the things, for example, that I'm doing together with Peter is trying to balance also a little bit this ecosystem of vaccine manufacturers, because it's ridiculous that Latin America has nothing, right? Where we have a whole continent that we have to wait for somebody to come and rescue and provide and sell vaccines to us, okay? With regards to the US, I think you all have now heard, about Operation Warp Speed. And Operation Warp Speed is, again, it's a partnership between the US government and the pharmaceutical companies, where, of course, the government gave money in advance for these 7 companies to produce in advance, even produce without even knowing whether it's going to work or not. And also they gave them money to do the very large clinical trials. So, each of them had to pay their own phase 1 and phase 2 trials. And now the government is paying for these large phase 3 trials. The other ones, Novavax, the Merck, the Sanofi, and the J&J, are also kind of coming behind the scenes. The rest of the world, unfortunately, the US has been not a very good player with the rest of the world, unfortunately. Initiative that is led by, again, the Bill and Melinda Gates Foundation, Gavi, that right now is not in the list of the US, but we are very soon going to be added in the vaccines that are going to be looked for the globe. There's a lot of information about that you may be hearing. Okay, when are they going to get approved? Is it going to be approved as an emergency authorization? Who makes the decisions of, when is a vaccine ready to be approved? So that is really based on what we call a blueprint or a product profile, which they have a threshold of basically saying, and I have to say the bar is relatively a low bar, which means as long as the vaccine can show that it's safe and that it can block at least 50% of the severity of the disease. So note what I'm saying. These vaccines initially, even though if they would be ideal, but they're not tested to only be approved if they prevent the infection. But they are looking for vaccines that they can prevent some of the infections, so in a way that it can reduce the severity of, if you were to be symptomatic, that you are not have severe symptoms, and that of course you don't die, and that you don't go to the hospital, and that you don't get into the intensive care unit. But they don't necessarily going to prevent 100% the shedding of the virus, or even the fact that you're going to probably still get infected. So very similar to the notion of how we use the influenza vaccine. Influenza vaccine, we know that some years can protect you 20%, some years can protect you 50, 60%. That doesn't mean that if you get exposed, you may still get the flu, but you clearly get a much lower severity of the flu, which is the key right now, right? The key to reduce the severity of the infections and of course, reduce the level of mortality. Same as the WHO, we have a lot of blueprints. In fact, all the big 3 pharma have now disclosed what are their blueprints. And there's some very intricate ways of how they do these clinical and safety development considerations. And when you ask me, when you hear, how can they do this so quickly, and this concept of acceleration and the timelines, it's really because they're trying to use innovative designs, but they're not really jumping over requirements or ethical requirements or safety requirements. So we hope that through clinical design, trial designs, through evaluating very specifically the endpoints, looking at, of course, the correlates of protection, we hope that probably by the end of this year, maybe the beginning of next year, we're going to start seeing, at least from those top 3 agents, pharmaceuticals, how they're behaving, these vaccines. The problem is going to be that we've been seeing more and more. In fact, this is a Pew Research article that just came out back in September 17, where even from May to today, the hesitance, even if we, all the work that we're putting into trying to, produce these vaccines and test them, a lot of people are saying that they're not going to use it. Which of course, is going to be very challenging, because what's the point of making vaccines and testing them and investing them and showing that, of course, they're safe and that they work, if then we have a lot of people that are not going to use it. And on top of that, they still don't use the masks, and they still don't use the, other health, public health methodologies. And the reality is that we did a study with Dr. Bruce Lee, which is a health economist, and you can read it in the American Journal of Preventive Medicine, that if you have a vaccine that is below this, 60, 50%, probably even 70%, we're going to still have to use a lot of the other measures of masks and distancing. Only when you start seeing that not only the efficacy of vaccines go above 70, even 80% of efficacy, plus the fact that you have high levels of coverage, meaning a lot of people use it, we are not going to be able to really interrupt or extinguish the pandemic. Now, again, I mentioned to you that many of the platforms that we are seeing are these platforms that are not used before in other vaccines, that sometimes they're also proprietary, but there are the ones that we consider ourselves the underdogs, the ones that maybe are coming behind the scenes. And as an example, of course, we have the vaccine that we developed, and I just wanted to put it in context, not giving you, much detail, but put it in context, because I think I have already I'm way over time. We already were working on coronavirus vaccines. Back in 2011, we started a program. It took us practically 4 years to develop a process to produce and test. Then we, of course, couldn't get any money, and we had to practically stall it. But the moment we knew that in January, this was a very similar coronavirus, believe it or not, because we had a lot of learnings in production, a lot of learnings in how we did the research, we really took only less than 5 months to redeploy a vaccine, to the point where what did we do? We took the coronavirus, we looked at the spike protein, we even looked at a piece of that spike protein. We produced it in a model which uses yeast, which is the same process to produce the hepatitis B vaccine. We did, some modest evaluation and testing. And then what we did is we now have transitioned all our learnings to, I'm going to skip this, to a company in India, which is called Biological E. They took it over from us around June. And as of today, which we're in October, they've already scaled, and they can make 1.2 billion doses of this recombinant protein. They already submitted their regulatory dossier to start a clinical trial in India, and probably that's going to happen in the next couple of weeks. And the good news is not only that it's affordable, and we hope that it's going to be safe and effective based on our preclinical data, we think so, but it's going to cost less than $3 a dose, which ideally would be well positioned to enter into this Gavi network of distribution. Plus, we can probably even have access to be able to even bring some back to the US in case some of the others are not effective. So what's the realistic timeline? Honestly, I think we hear all sorts of things in the news. Bottom line, based on some of the regulatory disclosures we've seen up to now, most likely not before the end of the year, we will not see any emergency authorization, probably between the end of the year and beginning of next year. How they're going to be deployed, probably initially in a very specific target of populations. And then slowly they're going to probably continue the studies to the point where we are going to be able to deploy even within the general population. So I think I'm going to stop there so that we can have at least 15 minutes or so for question and answers. And back to you. Not sure who I should send it to. I don't know if it's
I
you
can
should meet.
help mediate the questions from the chat. So someone asked, how does vaccine development factor in viral mutations?
How does vaccine development factor in?
Viral mutations.
Excellent question. Yes, absolutely, very important, because viruses, as well as practically any organism, including we humans, we mutate, right? Unfortunately, if the mutation is good, it may give an advantage to the pathogen or even ourselves. But sometimes the mutations, of course, are also deleterious, and that's one of the things that viruses may also use to not kill the host, right? Because remember, the pathogens, ideally, they don't kill the host because they want to be able to replicate themselves. With regards to COVID, we have seen, of course, there is mutations. This is not an RNA virus that mutates as bad as, for example, an HIV virus. But the mutations that currently people are tracking seems that they're not in areas within that spike protein that is going to really interfere its ability to bind that receptor and enter. Because remember, the virus needs to protect that, especially the receptor binding domain where it contacts the receptor. So right now, there's some mutations that are outside. We know that they're not essential. We know that, for example, neutralizing antibodies that are made by the people who are convalescent, or some of these vaccines, are outside of this. That said, very important, and when we look for the future, when we want to do for example, universal vaccines, maybe that can cross-protect throughout different coronaviruses, we need to pay very close attention of what is the epitopes within this pathogen that we're trying to target, such that we very rapidly adapt our sequences so that we can make vaccines that are not going to be obsolete.
Thank you, Dr. B. We have a few more questions. So why did you decide to develop the protein subunit COVID-19 vaccine over the other two common types?
Yes, so we actually, when we started the program with SARS, we joined a group of virologists, because in theory, we're not the virologists. We didn't really know much about coronaviruses, but scientists have already found in animal models that when you use the entire spike protein, or when you use these viruses in animals, it gave a little bit higher safety issues. People would, not people, but animals would react and would cause what they call sometimes immune pathology, exacerbate too much the immune response. Because remember, when you give the entire pathogen, you're not only making a response against what you want, but you're making responses against pretty much anything that is exposed. And so we, therefore, with this group of virologists, they had done some very important experiments showing that if you really narrow it and you focus your efforts into inducing an immune response to a very specific domain, which is the important epitope, you also remove the potential safety issues. We proved it in the lab, in mouse models, of course. To date, we actually, with some of the Chinese vaccines that indeed use this, whole concept of attenuated viruses, we do see that there is some reactogenicity, not still enough to say that they're going to be vaccines that are going to fail. In fact, they're quite protective. But we opted to always go for the least path of resistance with regards to technology. We wanted to keep it affordable. When you also make these vaccines, using the live attenuated viruses, you actually have to do them in, again, very high level biosafety, right? Because you have to manipulate the virus, grow the virus, then either use formalin or attenuate them by other means. And we didn't want to have to deal with the entire virus. There's so much knowledge now in the biophysics and biochemistry and the laboratory, that we can make vaccines synthetically that are easier to also do quality control, and you really remove any potential issues with regards to ethics and regulatory.
Our next question, someone asked, how would you approach and discuss the importance of vaccines with someone who is anti-vaccine?
You know, it is very interesting, right? I'm a firm believer that the majority of people who maybe have this thought that they don't believe in vaccines, it's just because they don't probably have the right scientific information or even the knowledge, versus the real hardcore, anti-vaccines that, even if you tell them anything, they're never going to believe, right? So I think the way that you approach it is first listen, right? Because most likely they have a behind the scenes story, like, oh, my kid, was born, and after I gave the vaccine, he was diagnosed with autism. It must have been the vaccine, right? Because people try to always put a cause of some effect. And they may not necessarily have the knowledge or really understanding why there is actually very strong scientific evidence that doesn't necessarily, occur. So I think it's, the dialogue, be good listeners. And then you as, eventually, scientists, professionals that, know a little bit the data, without not bombarding them with, very complicated scientific notions, is to just explain, right, what is the purpose of a vaccine, that in fact nowadays, they are so controlled, that you have to know exactly how you make them, how reproducible, what are all the components. There are so many checks and balances, and that the fact that, it's a risk balance, risk benefit ratio, right? It's probably worse to just have a Russian roulette that you're not going to get the vaccine, and God forbid you're going to get COVID, for example, if we were to have a vaccine. When the vaccine, we know that is going to really, protect you and reduce the burden of the severity of an infection. And I think you have to just, talk to them about what's the data available, and show that, in fact, we have saved so many lives by using these interventions. And the same with other interventions too, right? You have medicines, you have other technologies that really have changed the world.
Yeah, I think it's really interesting that even though there's a lot more transparency, there's a lot more people that kind of go against science now.
Right. And I think that's
so another person asked, what made you...
No, I was going to say that
Go ahead.
that is one of the things that, I also encourage you, that always try to dialogue, all the learnings that you get from the, of course, university, learn how to translate that to be able to converse with, someone who, of course, doesn't probably know anything about biology or biochemistry. And that's a skill, right? That's, how do you translate, very complicated scientific concepts to the layman terms. And I think it's a very important role for all of us scientists to eventually be able to do that.
Yeah, definitely agree. So another person asked, what made you personally want to go into this field of research?
So I grew up in Honduras, and this is where I am right now, FYI, if you didn't realize, and, we are a low-income country. Of course, I grew up, seeing a lot of these health disparities, social disparities. Of course, I liked, amongst the things in school, I tended to like more the biological sciences than maybe, I don't know, social studies or Spanish or, other areas. So it always draw me to learn, science. And I was fortunate enough that when I studied here, I'm a microbiologist by training, and a microbiologist not only learns about all the pathogens, but we are the ones who really tinker with the pathogen to try to identify how can you either detect it or eventually cure it, or eventually, of course, prevent it. And I think that's really what, to me, was my decision of, you know, initially I may have wanted to be the one actually treating the patient that had the disease, but then I realized that for me, you know, it was better to give the tools to the physicians for them to do better their job. And therefore I focus on more the research and understanding how I can provide the healthcare provider with the tools, which again are either how can they better detect it, or how can they better prevent it, or how can they better treat it. So that's why I decided to stay in the microbiology and specifically the research arena.
All right. I think we're going to do 2 more questions, or 3 more. But someone asked, given that so much focus has been placed on COVID and a COVID vaccine, how do you think this will affect NTD work going forward?
Well, one thing, it's true, you know, all the non-COVID related research, especially because of the fact that, you know, many of our labs had to close and many of the things had to be scaled down because of indeed the social distancing and even access to supplies and reagents. It has, of course, had an impact in the research. We may have been fortunate because, to be quite honest, we had a preferential ability of still going into the lab because we were really working on COVID research. And honestly, as we were doing the COVID research, we managed to be able to kind of like, you know, skip, you know, add under the table a little bit of outstanding experiments that we were doing with regards to our NTD vaccines. But, you know, in the field was hard. Like, for example, we have a trial, a clinical trial that we are doing in Uganda. And, you know, the physicians had a really hard time because they, you know, the participants couldn't come to the clinic or the physicians couldn't go to the field, right? So we have seen some, I think, across the board, some challenges. But now I think it's starting to, I think, retake, you know, slowly. Funding wise, absolutely right. You know that we have a tendency that we take from Peter to give it to Paul. And so, you know, we are now trying to also, you know, position the COVID indeed as a neglected tropical disease, meaning that COVID, seeing that it's such a systemic disease, that, for example, you know, someone who had Chagas disease, which is of course a disease that has cardiac involvement, and the fact that if that already predisposes you to probably get worse COVID, because of course, you know, you have cardiac disease, you know, we may be able to then apply for funds to say, okay, we want to study COVID in the context of Chagas disease, or, you know, how having a poor health immune system because you have all these intestinal worms, you know, affect, you know, having, you know, COVID. So I think eventually we're going to see some synergies. And I think the hope is that we realize that, again, R&D research and working in research and the reinvigoration of the importance of doing vaccine research, and of course any kind of translational science, has to have better business models and better investment strategies that are not also like this stop and go. Like we had the case of SARS, where we started, we got a grant, we worked on it, and when the grant ended, because there was no more SARS, nobody cared about it anymore. Instead of saying we should still continue it, because what if it were to come back, or what if something similar comes back? And hopefully that's going to change the landscape. Maybe we'll see what happens.
All right, and I think our last question for the day. Someone asked, when is research for a vaccine considered to be backed by justifiable data and able to pass the testing
Right. So this is when you, so the process of doing the clinical evaluation. So there's a lot of work, of course, that you do preclinically, but once the molecule is being, of course, produced by good manufacturing practices, meaning that you can actually use it in humans, you start these clinical development stages, which is, of course, the phase 1, phase 2, and then phase 3. And in a normal business scenario, right, you know, the phase 1 trials are usually very small. You look at the safety, you start looking at maybe some of the immunological responses. Phase 1s also help you to determine if you're going to do 1 dose, maybe 2 doses, maybe you want to change some of the formulations. That, and usually they're in what, 30, 40, 50 people. Then you go into these phase 2 studies, which you go maybe from the 30s, 40s to 200-300 people. You tweak a little bit, maybe the schedule and the design, and you start now looking at some of the correlates of, you know, protection. But that the ones that are really pivotal are, again, pivotal trials, which are the phase 3, which require, in depending on the type of disease, the type of course, of what you're trying to measure, you know, can go from a few thousand to even few, you know, tens of thousands, and even, you know, sometimes 30, 40, even 50,000 people. Those are still considered what we call experimental, but those are the ones that tend to be with long follow-ups. Right now, for example, one of the controversies is, when do you get the data to actually determine that you can stop these phase 3 trials? And right now the FDA said that you have to have at least 2 months of follow-up after you finish the scheme of vaccination. Normally the follow-up can be 6, 9, 12 months. And right now they're actually going to try to see what does the data look like after 2 months. But ideally they're going to continue the study 6 months, 9 months, and probably even 1 and sometimes even 2 years after. And that's why it takes so long, you see, to get the full-blown business as usual phase 3 trials. But then it actually doesn't stop there, because even after you, let's say, finish the trial, you give a license, every time you deploy the vaccine, you also have post-licensure surveillance. And so you can even have data that goes way beyond, you know, the licensure. And that's the reason why sometimes then vaccines are taken off the market, or vaccines are replaced by better formulations. Like we see this often with, you know, for example, the trivalent then becomes a tetravalent, then becomes a pentavalent, right? And it kind of replaces and switches, because you're improving, you're maybe even adding other things, and therefore you're making vaccines more certainly suitable for use. And that's why you know, it's a constant. So you really never end. But for licensure, you really have to go all the way through this phase 1, phase 2, and phase 3. And depending, sometimes, in fact, for some of the low middle income countries, there's even this so-called phase 4 clinical trials, which is the bridging between going from a you know, a high income population to a low income population, because sometimes you cannot assume that the health status of the people that you tested your vaccine in the US is going to be the same health status when you're going into a very underserved population in Africa, right? So they require you to probably do another study in those settings before you can extend the licensure into those low income country settings.
All right, I think that's all the questions that we have today. I'm going to hand it over to Shabneet, but thank you so much, Dr. B, for answering all those questions.
Thank you. Thank you for having me. I hope it was informative, and it really invigorated all of you to get into research. You know, you have lots of opportunities, I'm sure, within Baylor University. If you also are interested, there are opportunities at Baylor College of Medicine. So I'm going to, of course, send you all to talk to either Dr. Sanker or talk to Dr. Risk-Kloschmeyer, and they can also point you to some good opportunities within BU, and of course also outside BU.
Yeah, absolutely. Thank you so much again, Dr. Bottazzi, for being here and sharing your research with our burst members. Do you mind if we get a picture with you before you leave today? All right,
Absolutely.
awesome. If you guys want to turn on your camera so you can be a part of this, that would be great.
I don't even see myself anymore. I'm like, whatever.
All right, I'm going to take a few pictures because there's quite a few pages here, so just keep smiling. All right, here we go. Everyone smile. All right, I think I got everyone. Thank you so much,
Great.
Dr. Bottazzi.
Thank you. Thank you. Enjoy the rest of your afternoon, and I'm always available. You can find me within the Baylor directory, or of course, you can just email me also at Baylor College of Medicine.
All right, perfect. I'm going to share my screen again so we can go ahead and wrap up Research and Internship Day. Before we close, I just wanted to say a few acknowledgements of the people and groups who helped make Research and Internship Day possible this year. Of course, thank you, Dr. Bottazzi and the Baylor College of Medicine for being here. But also I'd like to acknowledge all of the undergraduate research presenters who shared their work this morning, all of the breakout session speakers, Dr. Klausmeyer from the Office of Undergraduate Research, as well as Ursa, McNair Scholars, the Career and Professional Development Department, as well as the Pre-Health Office and all of the pre-health organizations who helped us advertise today's event. And then last but not least, I want to acknowledge the Burst Special Events Committee, Annie, and all of the Burst officers for helping make today possible. Thank you so much. So with that, here is the QR code. Go ahead and take a picture and fill out the form so that you can get attendance for being here today. Several of the pre-health organizations are also giving points for being here. So make sure you list all of the organizations that you're a part of. And again, thank you so much for being at Research and Internship Day 2020. I know it's not usually how it is. It was our first virtual Research and Internship Day, but I think it went pretty well overall. So again, thank you so much for being here.